ranked abstracts in our session, and I'm very excited to see the results. Dr. Cohen.
Thank you very much for this very kind introduction. I'm very excited to be here, and I would like to thank you for this opportunity to present our work on behalf of our multiple co-authors and collaborators. Let's see if that's working. Okay, I'll just switch from here. That's fine. So our work is to aim to better understand metastatic breast cancer. That's perfect. Thank you so much. So metastatic cancer are cancer cells that had migrated from the breast. And that is a huge problem in the US and worldwide. Over 40,000 individuals in the US alone have metastatic breast cancer, which is one of the leading causes of death from cancers in women, and a huge problem for mortality and morbidity. So you can say that it's not the primary cancer that usually is the bigger concern. Now, we have treatments for ER-positive breast cancer that are very effective, but within the metastatic setting, drug resistance is almost invariable and frequently occurring. There is an urgent need to develop new therapeutic strategies for those patients who no longer benefit from these ER-targeted therapies. I should point out that knowledge of the molecular and genomic landscape of ER metastatic breast cancer is underexplored, as opposed to the primary ER-positive cancers, which we understand much better by now. Our current research is part of a growing effort by many research to start closing that gap. Now, our goal is really to understand evolved resistance. That is, to try and understand what are the mechanisms that drive the evolution of resistance in ER-positive metastatic breast cancer. And once we understand that, how we can translate that knowledge in the clinic. Better diagnosis and prognosis inform the choice of second line of treatment and ultimately durable control of the disease. As a brief overview of our approach and design, we had whole exome sequencing of 149 metastatic biopsies, and 44 out of these were matched with the pre-treatment primary. So I should just say that we are measuring both the exome and the transcriptome, and I'll go back to that point and elucidate that in a further slide. This is a first glance of the landscape. What we observe here is the significant genes that are altered in the setting of ER-positive metastatic breast cancer. Rows are genes, and they are organized from top to bottom by their significance, while columns of this matrix are samples that we had in our cohort. I would like to point out to few key observations that we have. First is that some genes that we find here are important also in the primary setting, as TP53, GATA3, and PIK3CA. However, since the landscape here, the selection pressures on the metastatic treated setting are different, we also highlight genes that may evolve not only for tumor genesis, but specifically to increase the metastatic potential and the endocrine therapy resistance of those tumors. I can point out to key potential drivers of the resistance phenotype: ESR1, RBB2, AKT1, KRAS, and RB1. And I will also point out some genes that may contribute to the metastatic potential. I should point out here that, switching gears, we wanted to make a head-to-head comparison. We wanted to be able to better understand how those tumors evolved. So if you have the metastatic setting, we wanted to have an earlier time point from the same individuals that are pre-treated. And this is, again, not to belabor the point too much, we can think about those as family trees. And we can think about the primary and the metastatic as two siblings that are diverged from a common ancestor. And then we can think about mutations. 1 second, please. As either shared, which means that that mutation appears both in the metastatic setting and the primary, or acquired. And those may play a crucially important role in that metastatic setting and drug resistance. Now, few key observations that we have. ERSA1 are mutated in about 23% of our cohort. Now, 14 out of 15 of those samples, when matched with primaries, were acquired, which emphasize the important role that these mutations may play specifically in the metastatic and drug-treated setting. Similarly, RBB2 mutations appear in 7%, and 5 out of 6 seem to be acquired in the metastatic setting. For both of these genes, other than being acquired and suggestive of their importance, these also have clear clinical implications, as finding those mutations may guide treatment choices for those patients. Last example, RB1 mutations that are found in only 5% of the patients. And again, the numbers are smaller. But since we have clinical implications, the very observation that those tumors evolve and that they have different mutations in the primary and in the metastatic may be important, because for clinicians, even if you know what are the alterations and the mutations in the primary setting, that may not be sufficient to guide treatment at the later time point of the metastatic setting. So the take-home message here is that tumors do evolve, and the metastatic setting is different than the primary setting. A final point to make here is that although it's very hard to assess the functional and phenotypic consequences of mutation in the clinical setting with patients, since we're sequencing both the exome and the transcriptome, that may give us a handle to assess that. So as a case in point, we have here a transcriptome map. On the x-axis, we measure ER activity by the transcriptome. On the y-axis, we measure cell cycle activity. Now, we're denoting samples by their mutation status. In red, these are ESR hotspot mutations, and in green, these are RB1 inactivation mutations. Now, what I want to point out to you is that the association between the transcriptome-based signature of ER activity and ESR1 mutations and the cell cycle activity on the y-axis and RB1 mutation may indicate that indeed those mutations are functional and they are having effect in those samples. Now, finally, I would like to acknowledge that we realize that tumors may evolve resistance through other non-genomic mechanisms, through epigenetic and regulatory mechanisms. Now, as long as those mechanisms leave a footprint on the transcriptome, the part of the exome and genome that are expressed, sequencing the transcriptome together with the exome may yield relevant insights into resistant states that do not derive specifically from exome mutations. So just to finalize and to summarize, implications for patients. So multiple clinically relevant alterations were identified in our study, with implications both for choice of next therapy, for clinical trial eligibility, and even for novel drug targets, which highlights the value of genomic profiling in the metastatic biopsies of ER-positive breast cancer. And I would like to finally acknowledge, again, the work from all of our collaborators, Nick Wagley, the senior author of this research, And if there is time, I'll take questions.
Okay, thank you. As we